Much, Mariana. Thank you so much to all the organizers, to BINA, to Nikki, to Vanessa, and for giving me this opportunity to share the work that we've been doing in my research group. And again, thanks for the very kind and warm introduction, Mariana. It is really an honor to be here to talk to all of you about different technologies that can be very useful in your research labs, including in core facilities. So we're hoping that the Laboratory for Fluorescence Dynamics gets converted into a core facility that can service not just UCI here in Irvine, but also the broader national community at large. So that's our goal and plan for the future. But just to give you a broad overview as to what my lab does, we do use imaging technologies that I've helped co-developed and Enrico's lab has developed for imaging, more advanced imaging microscopy techniques, including the phaser approach to FLIM, the number molecular brightness analysis, RICS, PCF, diffusion tensors. And all of these techniques can be applied to almost every field that you can think of. We worked with a diverse group of scientists over the many, many years at the LFD that used to be a P41 center. And these are just a few here I'm highlighting. So today I'm going to talk about metabolic imaging, as well as a new program that we developed called Mitometer, and I will touch on second harmonic generation imaging and what is our current research in the lab. So as you all know, and I don't have to go into, just in great detail, but we use multiphoton excitation to do most of our fluorescence lifetime imaging microscopy work and data collection. And the advantage of two-photon excitation is that it enables you to look deeper into tissues, specifically if you're looking at organoids as well. It just gives you that advantage. You can also do second harmonic generation. This is a non-linear process that occurs in the tissue, and I'll talk a little bit later about that. But two-photon excitation has a lot of advantages just due to the fact that you could just high probability of density of these photons exciting your electrons of the molecule that you're interested in seeing the fluorescence of. So in our study, we use the autofluorescence signature within cells. And every single cell in our body has nicotinamide adenine dinucleotide in the reduced form. And the reduced form of NADH is found in every single living cell. And I'm just going to give this as a background so you know what it is, what species that we're looking at in the cell. So NAD plus is not fluorescent, but NADH is, and that has to do with this dinucleotide. It consists of these two nucleotides joined through a phosphate group, which can be seen here in the structure, and a nucleotide contains an adenine base, while the other one contains a nicotinamide. So the reduced form of NADH is the result of accepting two electrons in a hydrogen that reaches a higher energy state, making it fluorescent. So you see here the difference between NAD plus and NADH. And the advantage of this is that NADH is found everywhere in the cell, but particularly it's mostly found in the mitochondria. You can actually see these mitochondrial structures really, really clear, specifically using two-photon excitation. NADH can be impacted by several biochemical changes, including pH, temperature, that has to do with their conformational and ionic changes and binding sites. But NADH has become a really useful molecule to study metabolism. It can act as a coenzyme in the redox reactions where NAD plus is it's the oxidizing agent that accepts the electrons. NADH is the reducing agent that can be oxidized, and it's really found in everywhere. You can also see it inside the nucleus, and we've done studies of NADH in the nuclear compartment in response to DNA damage. And so it can be really a useful biomarker for understanding what's happening within cellular systems. So to do our fluorescence lifetime imaging, we need a microscope, and most core facilities now have commercial microscopes in their core facility, which is fantastic and wonderful. And if you're able to excite, let's say NADH using multiphoton excitation, we use a TI sapphire laser. And because it's pulse, we have a time that we know exactly when the photons will be arriving. That's called the fluorescence lifetime, the excited state of the electrons as they're coming back down to the ground state. The time that it takes for that process to happen is in the order of nanoseconds. And that's really what we're measuring, is the excited state of those electrons. But of course, we can only detect it when the photon comes back and hits our photocathode, and we know exactly the time that occurs. So we need to synchronize this to our laser using some sort of card, a FLIM card. And so we've, in Enrico's lab, they've actually made their own FLIM card with an FPGA chip. And if you're interested in that, I'm happy to share that work in the chat later. So in fluorescence lifetime, you can actually use time domain, which I've already explained, using a very short pulse of light, femtoseconds, that when the intensity of the emission signal versus time done is recorded. When the time decays 1/e of the original starting value, that's about 36.8%. This is what we call the lifetime. Of course, that's very simple if it's single exponential, but if it's multi-exponential, and that could occur because the molecule can exist in different conformational states that then give it multiple fluorescence lifetime, and then it will have a multi-exponential decay. And that happens with really big molecules, let's say GFP or some of these other molecules. But if you're looking at, very small molecules such as fluorescein, rhodamine, they usually have a single exponential decay. So in our system, we're exciting molecules with a pulse of light at 80 megahertz, which means that we're stimulating 80 million times per second. This also means that the time between pulses is 12.5 nanoseconds. So we stimulate 1 pixel, and then we're measuring about 10 to 100,000 counts per second. And if you're exciting at 80 million times per second, this is about 80,000 times per second. So you're basically receiving 1 photon for every 1,000 pulses. So your photon density is very scarce. And we have to do that in every single pixel in our image, which means that we need enough statistics to collect enough photons to generate, let's say, the average time of arrival of those particular photons. So our contribution to FLIM is the transformation of this raw data, this would be in the time domain, into what we call the phasor space. And I'm just going to give a little brief overview as to what the phasors are. You can also use modulated light sources, and this is considered to be in the frequency domain. So if you don't have an ultra fast pulse laser, you can use a modulated light source. And then when the photons hit the photocathode, that signal is converted into, let's say, a modulated signal, but the amplitude of that signal is, let's say, dampened. So it's demodulated and there's a phase shift. And again, you can transform that raw data into what we call the phasors. And the phasors is a really nice way to graphically represent the fluorescence lifetime. It's a fit-free way to do it. So you don't have to worry about single exponentials, multi-exponentials. Basically, every single pixel in your image gets graphed onto this polar coordinate system. And then if you have very short lifetimes, they will be on the right side of the graph. And as they get longer and longer and longer, they go to the 0, 0 axis. And you notice here we have a vector called M, which is the modulation and change of phase. And it becomes easy to understand if you're doing frequency domain. But the advantage of the phasors is that we can calculate let's say, the fractional contribution of multiple lifetimes. So if you have free NADH, bound NADH, and some third component, such as oxidized lipids, the fluorescence lifetime of your pixel may fall somewhere inside this triangle, which would be a combination of 3 possible lifetimes, or they can fall somewhere along these lines, which will mean that you have a fractional contribution of just those 2 molecular species. Now, the phasors has kind of a long history. It's been used a lot in electrical engineering. This is an example of an AC current transformed into this rotating phasor or vector. But the first folks who used the phasors were Gregorio Weber, where here he derived that S and G coordinate that I showed you earlier on our lifetime transformation. And then Enrico and Dave Jameson were the first to actually make this graphical representation of the phasors with the S and G coordinate here that you see here. But we weren't the only ones using the phasors. Since the 80s, it has been used, and it's been kind of dormant, and it has been picked back up in the 2000s. So Tom Jovan and Andrew Clayton and Quentin Hanley used the phasors. They called them the 80 plots. Bob Clegg used the phasors. He called them the polar plots, and Enrico and I came out with phasor plot, and that was for the first time used for imaging for fluorescence lifetimes. So again, it's just, I'm going to actually skip through this because I have a very nice video that Lorenzo Scipioni made on the simulation of the phasors. But if you're interested on how this actually works, let's say that you collect a photon histogram. This would be the raw data that is collected on your imaging system. We have this sine wave here. This is this red wave. And essentially what we do is we calculate, we integrate the sine of the intensity from underneath the intensity raw data of our curve, and we multiply that by so we get some sort of value here of the sine. And you see this is the S coordinate 0.4. And then this number is done by dividing over the area of the black curve behind this. So you have the S coordinate in the phasor plot. Then we take the cosine transformation of this histogram here, and we repeat the same process, which is for the cosine, which is 90 degrees shifted. And we multiply we multiply this by the area under the curve, and we get the G coordinate. So now we have an S and G coordinate, and we'll have one spot here on the phasors. So this is an example of a single exponential decay because it's on the universal circle. For multi-exponential decays, the phasors will be inside of the semicircle. Now, some of you may say, well, what if it's inside the semicircle and I have two possible combinations of fluorescence lifetime? Well, in this formula here, you'll notice we have an n, which is the harmonic number. So for an n of 1, we would just plot the S and G coordinate for that pixel. You'll notice it falls right here. And now if we double that, so if we make n equal to 2, that would be the second harmonic. And this is just mathematical transformation. It has nothing to do with SHG or second harmonic generation. This is just multiplication factor here, where we double the frequency. And by doing that, what happens is you'll notice that the point now is a little bit shifted towards the 0, 0 axis. And how does this help us? Well, if you have a lifetime that's here, and we want to make sure that it's not a multiple combination of fluorescence lifetimes, we will increase the harmonic, so we will double the frequency, and now that point will be separated if they're truly multi fluorescence lifetimes that are occupying the same phasor coordinate. All right, so this will be just a demonstration. This simulation was done by Lorenzo Scipioni at the LFD, and you could see that the very, very short lifetimes, the lifetimes will be on this side of the axis. And then as the lifetimes get longer, they get demodulated and they go to the 0, 0 axis here. And then we can calculate the fractional contribution of two possible, let's say, fluorescence lifetimes here. So all of the possible fluorescence lifetimes that you have in your image can be plotted on the phasors, and this becomes a really nice way to look at this graphically on the phasor representation. Now, for our application, we are going to be looking at NADH as a metabolic marker, and to highlight some of the work that's been done in this area. And if you're interested in learning more about how you can actually get metabolism information from NADH, one of the nicest papers that's come out is by Melissa Scala, where she's shown in vivo multiphoton fluorescence lifetime imaging and the protein bound and free NADH in the precancerous epithelia. So one thing that we do know is that if free NADH goes up, that means that you have a lot more glycolysis happening in your cells. If you have more bound NADH, which gives you a very long fluorescence lifetime, we will have more oxphos. And the reason that you have a free to short lifetime for NADH is due to the conformation of NADH. So when it's in a free form, these two moieties of the ring structures overlap, they self-quench, so you have very short lifetime. And then when it becomes protein bound, this substrate opens up and the lifetime gets much longer. So in our work, we'll be using breast cancer cells and then imaging them using our fluorescence imaging system. And we're going to collect the raw data, which is the time arrival of the photons. And then under phaser, we know exactly where the free NADH, the pure species, the free NADH, we can buy this from Sigma, will make it bind to an LDH, lactose dehydrogenase, and the lifetime will get longer. So now we know exactly where the line of the fractional free to bound components are. And depending if there's more free NADH, will be more glycolytic or more bound. And you can see here the map of lifetime of NADH. Again, we didn't use any dyes. This is intrinsic autofluorescence of NADH. And you can see that these fibroblasts have a different metabolism than the breast cancer spheroid that you see here. This metabolic trajectory was defined by Chiara Stringari and Enrico Bertone in their work, where they showed that NADH in the mitochondria is more bound versus NADH that's being produced during glycolysis is more free. And this was verified using these experiments here, where you have more bound NADH. Adding potassium cyanide inhibits the electron transfer change, so you get more free NADH, forcing the cells to go to glycolytic states. And you can reverse the glycolytic state to a more oxphos state by using other chemical reagents. So this is just the same, and I'm just going to skip over this to get to some of the work that we've been working on. So we're interested in understanding metabolism and cancer progression. We know that mitochondria plays a big role in cancer invasiveness. There's a lot of papers that have shown mitochondria. Its structure can be a marker for aggressiveness in cancers. So I'm going to show you some work that we did that kind of fueled our interest in more mitochondria work. This work was done by my former student, Emma Ma, where we took collagen and we added cells, breast cancer cells, and we put them on very thick and very dense collagen structures, and we wanted to see if their metabolism changed. And so we also went on to look at other types of cells, including glioblastoma cells, and then really try to understand what's happening with the mitochondria. So in the first work here, this is just a summary of all of this work that was done, where we took non-tumorous cells. We put them on glass, which is the most stiffest material we can find. And then we added collagen, 3 megs per ml, 1.2 megs per ml, and then an inhibitor. So for the non-tumor cancer, non-cancer cells, the metabolism actually didn't change, no matter what substrate they were on. For ER/PR positive breast cancer cells, they also didn't really respond to the change of the environment. But for triple negative breast tumor cells, these are cells that are chemo-resistant. They're very hard to target. They don't have these receptors that are found in these other, let's say, less aggressive cancer cells. These are very aggressive cancer cells. What we did see is that they did change metabolism. So in the stiff material, which is the glass material, we did see a higher propensity for free NADH. So taking all of this, in the soft material, we saw that they were more oxphos. So that was really interesting, that in soft and pliable material, these tumor cells switch to a mitochondrial respiration. All right, in a collaboration with Stephanie Silitz and Dr. Sorabi, we looked at the environments that have kind of a native tumor environment in the brain. So these are glioblastoma multiformis spheroids that are embedded in hyaluronic acid gels in a stiff versus soft condition. And in the soft matrix conditions, there are little to no protrusions. So what you see here on day 1, you don't really see any protrusions, the stiff as well. And after day 3 and day 6, these cells start to invade the rest of the, let's say, environment, while the stiff material keeps them in place. So they did RNA, bulk RNA sequencing, and they looked at these heat maps that, and you can see here that the heat maps are for mitochondrial encoded genes or markers. And they saw that for both cell lines, so they had 2 different cells, the HK, these are patient-derived glioblastoma cells. The mitochondrial encoded genes, which are downregulated in the soft substrates, whereas in the stiff material, mitochondrial markers are being upregulated. So the same is true for both glioblastoma from the patient-derived cells, which means that glioblastoma cells overexpress proteins involved in OXPHOS in stiff hydrogels compared to soft hydrogels. So we also did this with FLIM imaging, and we found that there is a decrease in bound NADH, or that means that they're upregulating glycolysis in soft matrices. And that made us kind of nervous, because that seemed to go against what we saw earlier, where the more aggressive tumor cell lines were more OXPHOS, right? So what happened is in this overall data, for our RNA-seq data and the FLIM data in the bulk way, soft materials increases glycolysis, which is opposite of what we saw. But then my student was very sharp, and he said, wait a second, there are different populations of cells here in the tumor spheroids. Why don't we zoom into these very aggressive cells that are migrating out of the tumor? So when he did that, and you could see he zoomed into these, let's say, leading cells that are more, let's say, getting out of the tumor microenvironment. In the FLIM image, what he noticed is that the fractional contribution of bound NADH goes up. So actually in the soft material, there is a positive increase in OXPHOS, as we saw with our previous data. And these are only these invading cells where OXPHOS is going up. And that was really interesting, because what that told us is that with FLIM imaging, we have the capability of looking at single cell analysis and monitoring their metabolic profiles, as opposed to doing a bulk overview analysis, right? So bulk RNA-seq data didn't pick that up, but neither did our imaging here, because we weren't specifically selecting these cells that were protruding out of the soft material. So that correlated with our previous work. And now we actually turned to a collaborator. We worked with another great collaborator. This is Devin Lawson and Dennis Ma, her postdoc here at UCI, and they work on these PDX models. So they take primary cancer cells from human patients, they put them into mice, and then these cells are allowed to metastasize to different parts of the body. They now isolate these organs. They use flow cytometer to isolate the human cells, and then they perform single cell RNA-seq to see what genes have gone up when they metastasize to these distal parts. And so what they see is that mitochondrial OXPHOS genes versus, let's say, these are the micro metastatic regions, if the genes go up or down. And what they specifically look at in these metastatic sites in single cell RNA-seq is that we see an upregulation of OXPHOS, including many mitochondrial proteins. So what happens is at the tumor site itself, they see that they're more glycolytic, but when cells get to the organ, they're switched to more OXPHOS states. So what we decided to do is, the question was, can we downregulate OXPHOS? So maybe these tumors that are at the tumor site switch to OXPHOS, and then they go to the organs. So what if we are able to downregulate mitochondrial respiration? That way, no cells go to the organs. And we did this using a treatment called antimyosin A treatment or oligomyosin treatment. And we tested it with phloem. And we do see that using this treatment, we are able to switch the respiration from glycolytic to more to a more bound, so from a free NADH glycolate to a more glycolytic state, so more oxphos to glycolytic state. And then what happened is after we treated these cells, we injected them into the mice. We allowed those cancer cells to migrate to the distal organs, and then those cells were counted. And what you see is that using oligomyosin, we do see a downregulation of the number of cells that actually went to the distal site for both these two types of cell types. So inhibiting these mitochondrial processes greatly reduce metastatic spread. So overall, we see a metabolic shift in cancer progression in the glioblastoma cells. We did see that at the primary site, the cells are more glycolytic. And in the breast tumor cells, we see that in the primary sites, they're more glycolytic. And then when they go to the metastatic, when they metastasize, they're more oxphos. And that correlated the similar way with glioblastoma cells, where in the tumor site itself, they're more glycolytic and invaded cells are more oxphos. So this led to a question that we had is that mitochondria is doing a lot in the cell. Mitochondria is able to switch on and off these kinds of metabolic states. And what we know from the literature is, in particular, this example here where Caino and co-workers looked at the removal of a protein called synaptophyllin. And synaptophyllin is a stress-regulated mitochondrial switch of cell proliferation, motility, balance, and cancer. And it associates with mitochondria and microtubules to anchor the mitochondria and control their transport. So if they downregulate the mitochondria here, what happens is that if they use siRNA, it's a stop sign. So this synaptophyllin halts mitochondria from being transferred to different parts of the cell. But now if you inhibit this protein from being expressed, then the mitochondria are free to go everywhere. And so you see an increase in speed, and there's actually an increase of cancer invasiveness. So less synaptophyllin, the faster the mitos, they travel further and they're more metastatic. So we wanted to look at mitochondria transport in the cell to see if maybe mitochondria are being shuttled to regions of the cell that require a lot of ATP. So to do this, I had a student working on mitochondrial, let's say, transport, and it was taking forever. So she was like, this is going to be forever. It's going to be the end of my thesis. What am I going to do? And I said, let's use the photoactivatable RAC. This protein was made by Klaus Hahn and others. And this movie is not playing, so let me see if I can take out. Oops. Okay, here we go. Okay, this maybe it's not going to play. Okay, so we're going to go on to the next slide. Okay, so we were interested in having, there we go, to look at mitochondrial transport. And by photoactivating RAC, with this really ingenious way to do it, you can actually have the cell go in the direction that you want by photoactivating RAC. And so here what we see is that mitochondria, when we photoactivate this part of the cell, we do see a preferential, let's say, recruitment of mitochondria to these leading edges. And we did these on MB231 cells. These are the triple negative, highly aggressive tumor cells, and in MCF10A cells in non-tumor cell type. And basically, we don't see any preferential recruitment of mitochondria to these leading edges of the cell. And so that was interesting. We did a lot of analysis. And then when I talked to Klaus, he's like, oh, don't photoactivate RAC, just see what happens, wait the time. And so we did that. So we have photoactivatable RAC. You see there's a directionality preference when we do photoactivate RAC, and we see mitochondria is being recruited there. We don't photoactivate rack, there's still a recruitment of mitochondria. And these are NIH3T3 cells, these are control cells, and we don't see any directionality preference. And then the average speed of the mitochondria is increased here compared to just speed of mitochondria without doing anything in NIH3T3 cells. So I then got a new student once one student left, and that was the task for the student, is to follow these mitochondria over time. And so it was it was during, like, right before COVID, this was happening. We were imaging mitochondria, and we had a lot of issues. So first of all, there's not a lot of image software to track mitochondria. But you also get these kind of fuzzy looking images, as well as mitochondria that kind of overlap, or mitochondria that's not so visible in certain regions of the cell when you're doing a good cross section. So Austin, my former student, developed Mitometer, which is an automated approach to quantify the dynamics. He borrowed from astrophysics, where we are able to subtract diffuse background from the actual original image. And then by subtracting the diffuse background, we started to see more of the mitochondria in, let's say, in vision. And now, if we Gaussian filter and threshold this mitochondria, we're able to now generate a final image where we can really see a good contrast of the mitochondria from our original image here, which you can see has a very noisy background. So I don't have a lot of time to talk about this, but this algorithm is available on GitHub. So if you're interested in kind of a plug and play, if you have mitochondria movies, you can use Mitometer to track your mitochondria. And it's an automated way to do this. Essentially, we now have different types of diffuse background subtraction. We have a high background diffuse background subtraction that enables you to go from a very, very high, let's say, noise region to isolate the mitochondria, and very low noise region where we can use a lower diffuse background subtraction. You can actually see these mitochondria pop from the image here. And so we can also apply them to mitochondrial, let's say, movies from 3D spheroids. So we use these culture conditions to look at mitochondrial heterogeneity. We use normal epithelia. These are spheroids that are grown from patients that have had breast reduction surgery. So these are normal epithelia cells. These are our ER/PR positive cancer, non-aggressive, not as aggressive, but they're still tumors cells. And then our highly aggressive tumor cells. And these are also including from patient xenografts here. And so we were interested in determining what is the difference of mitochondrial tracking and movement in 2D versus 3D. And there's a lot of data here, but I'm going to summarize all of this data here. So in 3D versus 2D cultures, we see that mitochondria are slower. They're more directional. They're less robust of speed. They match the cell migration in 3D versus 2D. Triple negative breast tumor cells undergo more fusion events. Fusion enhances cell invasion. And if we look at triple negative and ER positive cells, the network, because we have different parameters we can not just track mitochondria and get the means for displacement, but we can also take the structure of the mitochondria and evaluate if they undergo more branching, if they're less branched, the axis, all of the features of the structure, including if they undergo fission and fission events. And with the ER versus the ER/PR positive versus the triple negative cells, we see that the triple negative cells are more networked, they have lower membrane potential, and they're less rounded. So in general, we see that normal cells look very similar to triple negative breast cancer cells. And it's really hard to target them, mainly because of their phenotypes look very similar to the breast tumor cells. So here's an image, just wanted to show you what the tracking in spheroid looks like. These are glioblastoma multiforme spheroids embedded in hyaluronic acid in soft conditions and in stiff conditions. And then the differences here in terms of their phenotype and motion is that mitochondria can travel much faster in the soft material versus the stiff material. They're more directional in the soft material, and they're less branched in the soft material, and their major axis length is much longer in the soft material. So again, just to summarize, we have now a lot of different types of parameters for mapping these mitochondrial structures with this. And from what we gathered from our metabolic imaging, we saw that soft matrices induce glycolysis. So overall, I showed you this work where FLIM is a powerful tool to measure metabolic state in cells. We saw that these triple negative tumor cells exhibited a decrease in fraction of bound NADH, indicating more glycolysis when in stiff material, but in soft material, they're more oxphos. And then we were able to use glioblastoma cells and triple negative breast tumor cells based on the PDX models to also see that metastatic cells seem to be more oxphos, and we can shut down the oxphos pathway and we see less number of cells going to the distal organ sites. We've developed mitometer for mitochondrial phenotyping and phenotyping profiling in breast cancer cells. And of course, the future work would be to maybe use a more multiparametric approach to understand cell behaviors. And to some extent, and for the last bit here, I'm just going to highlight some of the things that we've done to go into the space of using more of multiparametric analysis of what we can do with our imaging approach. So, of course, we're in our model system, we're using breast cancer. Breast cancer is one of the most common kinds of tumors. Specifically triple negative breast tumor cells. And we want to understand what is happening at the metabolic state, understand what's happening in the, let's say, intracellular lipid, because we can actually label these lipid droplets, and then what's happening in the extracellular environment to see how they change as a function of time. And so we are taking these tumor spheroids and embedded them in collagen matrices. And so this is our, let's say, preparation of our spheroids. We make these spheroids with round bottom wells, and we embed them in collagen. So we're able to look at the second harmonic signal for collagen, let's say, restructuring by these tumor cells. And of course, we're using multiphoton excitation. And as I mentioned already, we're using FLIM to look at NADH, and then that's the metabolic state. We're using hyperspectral imaging to enable us to label the lipid droplets. We are using Nile red, and then we transform our emission signal into the polar coordinate system, the phasers. So it's not FLIM phasers, but it's spectral phasers. And this work of the spectral phasers was originally done by Hans Gerritsen and Federoni, his student, who's now at UC Davis. And in addition to this, because we have the multiphoton excitation laser that we're using, we have the advantage of looking at the material, which is the environment, the collagen itself. We use second harmonic generation, which is a nonlinear optical process that involves the generation of light twice the frequency and therefore half the wavelength. So whatever wavelength we're using, if we're using 800 nanometers, we just look at the 400 peak to look at the second harmonic image of collagen structures. And this occurs because the intense light interacts with the collagen structure that has a nonlinear, let's say, nonlinear symmetry or asymmetry that occurs in collagen. So by getting this image, we don't just look at it, we actually analyze the collagen image using PLIX, which is known as phaser local image correlation spectroscopy. And essentially, it's just an image correlation analysis that's transformed into the phaser space that gives us the profile of the size and density of that collagen structure. So here's just some images looking at multimodal signatures of these triple negative breast cancer cells compared to the MCF10A cells. These are the control cells. And we use fluorescence lifetime, spectra, PLIX, and then of course the intensity. So this is our let's say parametric approach. And we can, with FLIM, we're looking at metabolic signatures. With spectral imaging, we're looking at lipid polarity and lipid droplets. With PLIX, we're looking at fiber thickness. And of course, we get all of the intensity image here. And some of our data already is showing that after a certain number of hours of these spheroids, we see that the control cells do not invade into, from the spheroid itself, but the MB2 triple negative tumor cells invade into the collagen. And then in the MCF10As, in terms of distance from the center, you can see that they travel much further. In what's happening with the FLIM NADH image, you see that MCF10A cells have a higher fraction of NADH that's bound compared to MB231s that have more free NADH. But as they start to migrate out, invade into the material, you see that their OXPHOS state goes up, similar to what we saw earlier. Also in the Nile red signature, what we're seeing is that in this case, the MB231 spheroids have yellow Nile red emission, so lower lipid polarity compared to the MCF10A spheroids. And the triple negative MB231 spheroids decrease lipid polarity as a function of time and distance from the center at 2 hours, while at 24 hours, the core in the compartment while at the 24 hours, the core is the compartment with the lower polarity. What happens with the PLIX, with second harmonic generation reshaping, we see that MB231 cells are reshaping the collagen more than the MCF10A spheroids due to the cells migrating and invading the surrounding. So in summary, we're able to multiplex different let's say, regions of the spheroids and look at each cell. We're able to look at the changes of lipid and polarity of lipids and, let's say, lipid droplets and see what is happening to the surrounding environment. So we believe that these kinds of ways to multiplex imaging techniques will improve the knowledge of what's happening to the metastatic cell invasion, specifically in cancers. So finally, I'd like to acknowledge everyone in my research lab and the Laboratory for Fluorescence Dynamics, which now I'm directing. Professor Enrico Bertone has retired, so I'm inheriting all of the responsibilities of the LFD here. And again, our plan is to turn it into a national resource for everyone who wants to get trained. And so I want to thank Austin, who developed the mitometer, and Lorenzo Scipioni for a lot of his work, and Giulia, who did all the spheroid work in this, and all of our collaborators and research funds. And finally, I just want to leave with this final, let's say, PowerPoint slide on our workshop. As Nikki mentioned, we are having our 18th LFD workshop in October, and you're welcome to join us. BINA provides a very kind professional development funds to be able to go there. So if you're interested, you can just click, get some information from these QR codes for the link. So thank you so much for your attention, and thank you so much for having me today.
Thank you so much, Michelle. That was a wonderful talk. Very, very interesting. I mean, I wanted to start first if anybody has questions from the audience. I have a few, but I'll open it first for the audience. If you can unmute yourselves if you want, otherwise you can write it in the chat if you feel more comfortable. If not, no. Okay, can I start? I had several. So have you ever tried to, so the first one I imagine more on the applied biology. How different are the mitochondrial profiles for different types of cancer? So you mentioned breast cancer and different forms. So for other types of cancer, do you see a lot of differences?
I think to some extent, yes. I think that even within the non-invading cells and the ones that are just in the tumor itself, we do see distinct differences. So we've seen that if mitochondria respiration is low, the mitochondria are very small, they're not elongated, they have less branching. So yeah, their phenotypes can change depending on the cell type and depending within the cell type, maybe in tumors specifically, because there's so much heterogeneity across cells that they can vary depending on what function that they're doing. So yes, we haven't profiled every single type of cell, but we've now looked at neurons, for example, in Alzheimer's disease models, and we do see that there's distinct differences. And we're starting now to look at genetic transcription factors that are important during stem cell, let's say stem cellness in cancer and chemo-resistant cells. We do see that perhaps in those types of cells where if you knock down a gene that's important in a particular process that affects metabolism, you do see a change in metabolic or in mitochondrial structure. So yes, it's very fine, very minor, but there are changes.
Okay, that's interesting. There's a comment on the chat. Some people have been trying to use flame as a way to identify cancer tissue due to glycolytic metabolism compared to surrounding normal, e.g. in guided surgery. Do you think that this will be challenging to do in secondary tumor sites given the shift towards OXPHOS? That's Jason Smith.
Yes, thank you, Jason, for that question. That's really great question because you know, some of the questions that I had looking at some of that data in terms of real-time surgeries is that when you cut a part of the tissue, you have inflammation. So metabolism changes just from inflammation. So I think that machine learning can actually be a good way to try to understand the changes of metabolism in this process of, can I tell the difference between just inflamed cells and tumor cells? And I think that we've got to develop not just one way of looking at this, not just maybe a FLIM signature, but maybe a multiparametric way of looking at this and try to use machine learning to identify which are the regions that are inflamed, which are the regions that are cancerous tumors. But from the data, it looks pretty promising that there are separated, but it still worries me that just you know, the inflammation itself can modify the metabolic profile in the NADH signature. So maybe looking at NADH with FAD, this is something that Melissa Scala has been working on, and many other folks at Tufts University are also looking at.
Great. There's another question, which I will link to one of mine. So you started mentioning that there was a difference what you detected in 3D spheroids versus 2D models. And so I was about to ask how this differs. So if you tried it in animal models, and so Maria Fernanda Forni is asking, she says, absolutely beautiful work. Do you think it would be possible to image the mammary tumors in vivo using these techniques, fluorescent probes? How much that's a different question. How does the metabolic niche change in terms of nutrient availability over time, and how does that impact metastasis?
Yeah, that's a great question. Looking at metabolism in the actual mouse models in real time, that would be ideal, honestly. In vivo, as a function of time, we've done a little bit of work on that, but obviously a lot more can be done in that space, in vivo systems. Fluorescent probes in vivo, that's where there's some challenge, because of course you want to make sure that you're using probes that either are FDA approved if you're thinking of the clinical applications to those. But certainly I think that multiplex, I think also another way to look at it is hyperspectral imaging. I think we're not making enough use of hyperspectral imaging and multiphoton imaging for autofluorescence signatures. And I think that could be a way in for clinics and for in vivo monitoring, because it's not as, let's say, complex as a FLIM system. So I think there's a lot to do for young people here in the audience who are interested in continuing this work. Yeah, but surely, Maria, that would be ideal. Yeah.
Absolutely. Yeah, you could link it to, I imagine, intravital microscopy.
Yes, yes.
I don't see another question, but interrupt me if anybody. Okay. Nikki is saying individuals with dense breast tissue have a greater risk for breast cancer, and I assume this was because it impacts detection, but it looks like density may also play a role in how the cells are behaving in vivo too. Would be great if some of these imaging techniques could be done in vivo in people.
Yes, absolutely. Thank you, Nikki, for bringing that up. Right now, the system that I know of, Hila Balu at the Beckman Laser Institute here at UCI has been working on a clinical 2-photon lifetime imaging system, but so far for just surface level. So for example, looking at melanoma and skin cancers, but certainly there's a lot more work that needs to be done in looking at more deeper tissues, such as the breast. And of course, maybe multiplexing technologies that already have existed, looking at oxygen, deoxygenated and oxygenated oxygen with tomography could also help to understand the tumor itself, and with second harmonic generation, at some point, if we're able to go deep enough into the tissue to look at the density of that, the collagen. But that's a great question.
I don't see another one yet, so I might ask one of mine. In terms of the multiparametric approaches, has anybody or have you done or thought of combining? It's a naive question. Combining AFM with FLIM.
I have not. I haven't seen that. Maybe somebody in the audience, maybe they know of a paper combining AFM with FLIM, but not specifically the combination of the two. Yeah. Mm-hmm. Yeah, we were just talking about AFM yesterday on trying to understand the membrane tension, right? So you can measure membrane tension, but there's multiple applications for AFM.
Yeah,
that would be cool to multiplex that as well.
Yeah, I mean, it came to mind there are studies showing exactly like the difference in the viscoelastic properties of the cancer cells at the edges of tumors that are going to metastasize and so on. So it just came to mind when you were talking about the heterogeneity in the tumor. Any other question? I think if not, I have one more. Can I ask one more? Well, I mean, we're close to time, but yeah, it's up to everybody. Yeah, I can contact Michelle afterwards. It's okay.
Hey, it's fine. If people have to go, they can go. You can still ask your question. Anybody that wants to stay can stay.
Okay, one more from me anyway. So I'm just interested. So in infectious diseases, intracellular pathogens, have you done any of that? So I'm interested in how difficult or easy it would be to do this phenotypic profiling of the mitochondria, because in many cases they some of the pathogens hijack host stuff, including mitochondria. and then start building their own from there. So that would be, I think, something.
That would be really amazing to look at. We have not looked at that. But yeah, that would be really super amazing. Specifically some of the viruses, right? I'm not sure which, there's some viruses that do hijack some of these machineries, these transport machinery. So it would be really interesting to see that real time. Yeah.
Cool. We should talk about it.
Yes, for sure.
Okay.
Thank you for those questions. They were great.
No, thank you. I think with that, if there are no more questions, thank you so much again, Michelle. It was wonderful to have you. And
thank you for having me. Thanks to all the audience here. It's great to see, some of you in here. So thank you.
Thank you so much, Michelle, for the great presentation, and thank you everyone for joining us. Next week, Mariana and Ivy will be presenting about their experiences at the LFD course. So please do join us for that. And then we hope you have a wonderful summer. Thank you very much.
Thank you.
Bye, all.
Everybody.